Hello everyone, we're group 13 and we will be presenting on the small intestines. My group members are Jada Smith, RJ Smith, K. Marie Clark, and Danny Plummer.
Objectives. By the end of this presentation, we would have hoped to achieve 1. The identification and description of the structural modification of the wall of the small intestines that enhance the digestive process. 2. The explanation of the roles of the various cell types of the intestinal mucosal glands. And 3. The route by which bile is secreted and released into the small intestines.
The second part of the small intestine is the duodenum, which is about 2.5 meters long. It contains muscular flaps and tiny finger-like projections called villi, which helps absorb nutrients produced during digestion. And finally, the third segment of the small intestine is called the ileum, and it is roughly 3 meters long. It ends at a pouch called the cecum, which is a part of the large intestine. The ileum's primary function is to absorb any remaining nutrients from digestion, primarily bile acids and vitamin B12. The walls of the small intestines are made of several layers. Firstly, there's the mucosa, and this is the innermost layer where the nutrients are absorbed into the bloodstream. It is lined with finger-like projections called villi, which increases the surface area for absorption. Secondly, there's the submucosa, and this layer contains nerves, blood, and lymphatic vessels, which supports the mucosa and helps with the transportation of the nutrients that is absorbed. Thirdly, there's the muscularis externa. This layer is responsible for the movement of food through the small intestines. It consists of muscles that contract and relax to push the food along the digestive tract. And finally, there's the serosa, or sometimes called the adventita, and this is the outermost layer, and it provides protection and supports the small intestines. It also helps to keep the intestines in its place within the abdominal cavity. Together, these layers work harmoniously to aid in digestion, absorbing nutrients, and facilitating the movement of food through the small intestines.
The structural modification of small intestinal walls. The wall of the small intestine has structural modifications that aid in its digestive process. These are 1. Brush border cells. The brush border cells are the villi and the microvilli. Let's start with the villi. These are finger-like projections that increase the surface area of the small intestine, allowing for better interaction between chyme and digestive enzymes. This increased surface area enhances both digestion and absorption processes. Next up, we have the microvilli. The microvilli covers the surface area of the villi, forming the brush border. This brush border contains various enzymes crucial for breaking down carbohydrates, proteins, and nucleotides. Microvilli further increase the surface area, aiding in the diffusion of digested nutrients into absorptive cells. The circular folds, or the plicae circularis, are folds you see within the lining of the small intestine. They serve to enhance absorption by increasing surface area and slowing down the passage of chyme by causing it to spiral rather than move in a straight line as it progresses through the small intestine. This slowing down allows for better absorption and interaction with the intestinal lining. The crypts of Librecon, or the intestinal glands, are located between the villi. These contain various cell types, including absorptive cells and goblet cells that secrete mucus. Pannette cells found here play a role in regulating intestinal bacteria via lysosome secretion. Additionally, enteroendocrine cells release hormones crucial for digestion. Myenteric plexus. The myenteric plexus controls movements within the small intestine. It orchestrates segmentations which mix chyme with digestive juices for absorption. It also coordinates migrated motility complexes, a cyclic recurring wave pattern which help move chyme along the intestine. Brunner's glands. Brunner's glands are located in the submucosa. These glands secrete alkaline mucus to neutralize gastric acid in chyme, creating a favorable environment for enzyme activity.
Mucosal gland cells of the small intestines. Numerous cell types found in the intestinal mucosal glands are crucial to gut health maintenance and digestion. The intestinal mucosal glands' diverse cell types work together to aid digestion, nutrition, absorption, pathogen defense, and maintenance of gut health. Goblet cells, Paneth cells, enterocytes, enteroendocrine cells, and stem cells are among these cell types. Goblet cells. Goblet cells are essential for protecting and moisturizing the body's epithelial surfaces by secreting mucus and a dense gel-like substance. They are present in large numbers in the small and large intestines, creating a barrier against digestive enzymes, toxins, and pathogens. Mucus aids in smooth food passage, supports healthy bacteria growth, and manages the hydration of the intestinal lining by trapping and retaining water, crucial for maintaining proper digestive system functions. Enterocytes. Enterocytes, the most abundant cells in the intestinal lining, absorb nutrients from digested food and transfer them into the bloodstream. Their structural characteristics enable efficient nutrition absorption, water absorption, barrier function, and hormone and enzyme release. They absorb vitamins, minerals, fats, proteins, and carbohydrates from food. The microvillus on their surface increases surface area for nutrient absorption and contains transport proteins. Enterocytes also help maintain fluid equilibrium by absorbing water and preventing dehydration. They also build a strong barrier to keep toxins, infection, and harmful substances out of the bloodstream, ensuring gut health and protection from pathogens. Paneth cells. The small intestine also contains specialized cells called Paneth cells. In the immunological defense of the gut, these cells are essential to defend the intestinal lining from dangerous germs and pathogens. Paneth cells secrete a variety of antimicrobial peptides, including lysozyme and defensins. They further help to maintain intestinal homeostasis and control the makeup of gut bacteria. To preserve a healthy gut environment and guard against infections, Paneth cells are necessary. Enteroendocrine cells. Enteroendocrine cells found in the gastrointestinal tract lining regulate digestive processes by releasing hormones in response to stimuli. They regulate appetite, satiety, pancreatic enzyme secretion, and gut motility. Hormones like ghrelin and peptide YY regulate hunger and fullness, while gastrin stimulates gastric and secretion. Secretin and cholecystokinin regulate pancreatic enzyme secretion for digestion. These cells also influence food movement, nutrients absorption, and movement. Stem cells. Stem cells in the digestive system are crucial for the renewal and repair of the intestinal epithelium, ensuring epithelial regeneration, tissue repair, and homeostasis maintenance. They divide and differentiate regularly to replace damaged or worn out epithelial cells, maintaining the intestinal barrier's integrity and nutrient absorption. In response to injury or inflammation, stem cells can quickly expand and differentiate into specialized cell types, repair damaged tissue, and ensuring epithelial cell turnover and tissue homeostasis.
The route of bile secretion. To help with fat absorption and digestion, the liver produces bile, which is then kept in the gallbladder and discharged into the small intestine. The following is a summary of the procedure. Step 1. Liver secretion. The liver cells are always producing bile. Bile is made up of water, electrolytes, phospholipids, cholesterol, bile acids, and bilirubin. Step 2. Bile ducts. The liver intrahepatic bile ducts are a network of tiny ducts that transport bile that is made in the liver. Larger ducts are formed when these conduits eventually combine. Step 3. Common hepatic duct. The common hepatic duct emerges from the liver after merging bile channels. Step 4. Cystic duct. Connecting to the gallbladder, the cystic duct joins the main hepatic duct. Bile is concentrated and stored in a tiny organ called the gallbladder. Step 5. Storage in the gallbladder. Bile is concentrated and kept reserved in the gallbladder in between meals. The gallbladder then releases bile when the body needs it for digestion. Step 6. Common bile duct. The common bile duct is formed when bile from the gallbladder enters the common hepatic duct after passing via the cystic duct. Step 7. The sphincter of Oddi. The duodenum, the first segment of the small intestine, is joined to the common bile duct via a tiny orifice known as the sphincter of Oddi. The amount of bile that enters the small intestine is controlled by this sphincter. Step 8. Enter into duodenum. Hormonal signals causes the sphincter of Oddi to relax when food, particularly fatty food, enters the duodenum. This facilitates the passage of bile from the common bile duct into the duodenum, where it helps break down and emulsify lipids. By taking this path, bile is efficiently released into the small intestine to aid in the breakdown and absorption of lipids through the digestive process, stored and concentrated in the gallbladder and discharged from the liver. The end.